All right, Bismillah, Assalamu alaikum. So I'm going to be looking at one of the links that I sent that I just listed in the previous slide, and this website shows very nicely the concepts of enzyme specificity, how enzymes can bind to specific substrates, as well as the binding model that we've talked about earlier, the lock and key versus the induced fit model. Right. So I'm expecting all of you to read. Please read whatever is here on the left-hand side. It's really important. Now, what's showing here is a picture of chymotrypsin, the enzyme that we're looking at, and it's got a substrate bound in it, and they're highlighting few of the um, enzymes that are involved in catalysis and the enzymatic reactions. So you have the histidine, the aspartate, and you have the serine here somewhere. Okay, so uh, after navigation through this section, you will have a clear idea of various factors that influence substrate binding, as well as current models of enzyme and substrate binding. Okay, so again, this is showing you the binding pocket of chymotrypsin and the three amino acids that we know are critical, which are aspartate, shown in green here, histidine in purple, and serine shown in this orange yellow color, right? And what they're showing you also this hydrophobic pocket in blue, and showing you in white is the peptide substrate, which is in the active site, and you can see the aromatic ring here, which is very nicely um, uh, resting in the hydrophobic pocket, and that's what's driving the substrate. So the text says the behavior of an enzyme has its physical basis in the three-dimensional structure of the active site, which is part of the protein that interacts with the substrate. And then go down. So what they're saying is complementary fit between the substrate and the enzyme accounts for the enzyme specificity. And this notion of complementary fit is just like a lock and key, right? So that determines the enzyme specificity. If a key cannot fit in the lock, then that substrate cannot be acted upon by the enzyme. So as an example, we know the chymotrypsin catalyzes the same reaction as other proteases like trypsin. So trypsin and chymotrypsin and there are many other proteases. They all catalyze the same reaction, which is breaking this peptide bond. This is C double bond O NH peptide bond, C double bond O NH peptide bond. However, what they're saying is, but chymotrypsin acts or recognizes only peptide bonds next to aromatic amino acids. So if you have an aromatic amino acid at the C terminal of those peptide bond, that's what chymotrypsin cuts in. So this is again a peptide bond, but this is not aromatic. So chymotrypsin will not work on this, but it will work on this. So that's what the text says. Chymotrypsin catalyzes the cleavage of polypeptide chains adjacent to the C terminal side chain of aromatic amino acids. This, if you have a positively charged amino acid, this is what trypsin works on. So trypsin does not work on hydrophobic, but trypsin works on a positively charged amino acid like lysine or arginine, and chymotrypsin has this aromatic specificity. Okay, so click on the amino acid with the aromatic side chain. So this is not aromatic, this is not aromatic, and this is aromatic. Okay, now what's shown here is this blue. They're showing you, a typical given hypothetical enzyme that has amino acid side chains, which are shown in this alanine and isoleucine, and then these are aspartate and amino and water and another amino group here. So these are all uh, a protein, but what's shown in the substrate is in red. So this red guy is a substrate, right? So they're showing you a substrate bound to a binding pocket in the protein. And what they're saying on the left hand side, that non-covalent interactions between substrate, which is the red guy here, and the active site, which are all the amino acids that are shown here, side chains are shown here, contributes to the affinity and specificity of the binding between the enzyme and substrate. Examples of these interactions that we know are hydrogen bonding and ionic charge interaction, as well as hydrophobic interactions. Right? And these interactions are what gives you the initial binding of the substrate. Right? So they want you to. So if you have this is a charge interaction. This is be hydrogen bonding between the substrate and the active site. This is again a charge interaction, and this would be the hydrophobic interaction because you have nonpolar groups. Okay. Uh, Now they're saying you what do the amino acid side chains next to the trypsin cleavage side. Now again they're showing you this is a trypsin substrate. This is a trypsin substrate, meaning this is a substrate that trypsin can work on, can cleave here. This is a trypsin substrate that can cut here. And they're asking you what is common, what is this and what is this. So these are both, they're not aromatic, they're not hydrophobic, and they're not acidic. Acidic, remember, will have COOH. So this is the basic, they have basic side chains, right? Not acidic. So this is basic. Okay, now given that the substrate set residues are basic and positively charged, which complementary amino acid would you expect trypsin to have in the active site next to the substrate? So if the substrates are positive, right, then the amino acid side chains that are interacting with them must be the opposite, which are acidic. Okay, and that's what they're showing you. This is trypsin. In trypsin, they're showing you the substrate. This is the substrate that has an amino charged substrate, and the active site will have a negatively charged amino acid in trypsin, which is the carboxylate amino acid. That's what they're talking about in the previous slide. Here, trypsin has a hydrophobic binding pocket. That's where the substrate with the hydrophobic side chain will fit in there. Okay, I think we'll just stop here for now. Let's see.